propose new features for the Caver. Then regarding the user support, there are also 2 mailing lists where we will be happy to answer any questions about Caver usage and help you to set up your system, the calculation for your system. Okay, regarding the relevant papers about the Caver, so the main paper was published last autumn in Post-Computational Biology, and there you can read about Caver algorithm, its performance, and its application to some biological systems. The second paper in Biotechnology Advances deals with tools for analysis of tunnels and channels in general. And the last recent review provides a catalog of very important dynamical parts of the tunnels, so-called gates, which governs the usage of these tunnels in various enzymes. Now to the Caver algorithm itself. Basically, it's search for the tunnel from some defined starting point, which is generally in some buried cavity, and it searches for its way outside. The tunnel is something connecting the buried cavity with surface of the protein. Caver represents a protein structure by its Voronoi diagrams and identifies the cheapest path to the surface, which generally corresponds to the shortest and widest tunnel, something like that. And it simultaneously finds all possible pathways from the inside to outside, so you can get more tunnels at once. It's important to mention that for the robust analysis of tunnels, it's best to search not only in a single static structure, but to use an ensemble of structures, which has 2 advantages. It allows the detection of more tunnels, and maybe more importantly, it then helps distinguishing relevant tunnels from some artificial irrelevant ones. The ensembles of structure you can obtain in various ways. We prefer molecular dynamic simulation, but you can use NMR, you can employ multiple crystal structures, normal analysis, or just try to query a few databases with some dynamics ensembles. So once we got this ensemble of structures, the Caver is used to identify tunnels in each snapshot, or maybe you can call it a structure. So you get a set of various tunnels. Then these tunnels are merged on a single structure, and Caver clusters them to the individual tunnels. This provides you actually the relationship or identity of corresponding tunnels across all the structures or the analyzed structures. And if you have MD simulation or something like that, then you even can talk about the dynamics of the tunnels itself. Or if you have just a simple set of static structures, you can still consider the variability in the tunnels. So now more to the Caver itself. So from the technical point of view, it's a Java-based software, so you have to run it using the Java. You have to use this really long command where the switches are to specify the maximum memory you can use. Then you have CP and jar switches. They relate to the installation of the Caver, so they are pointing to Caver files depending on where this is installed. Probably SB Grid will guide you on that. Then you have Caver home, which is important. It contains a bin folder, and in this folder there are some templates you may want to modify at some point. Then PDB, it just points to the folder where you store your PDB file or files of structures you want to analyze. Then you have a switch showing or pointing at the Caver configuration file, and finally the out switch, which tells the Caver where to store the results. So what are required Caver inputs? So it's already mentioned a configuration file where you have to specify a starting point for the tunnel calculation, and you can adjust many parameters. Then there is a bin folder containing the atomic radii and templates for visualization. Both default configuration file and this folder are provided within the Caver package, so you either have it or you can download that simply from the web pages. The last input is a folder with these PDB files, and it's important that the files are aligned beforehand, since Kver do not provide some structural alignment. And also, you should notice that actually the name of PDB file is used to initialize the random number generator in Kver. So if you want a fully reproducible result, you have to maintain the same name of the file. Okay, now if we provided all inputs, so what can Kver give us? So the results from the Kver calculations are stored in a user-specified folder. There are summary files, log and warning files, and data folder, which is just for Kver, and 3 others which we will discuss later. In summary.txt, there are overall characteristics of all calculated tunnel clusters. You have the ID of the cluster, which will allow you to map this information to the visualization. Then you will get the idea in how many structures was the tunnel identified, what was the average radius of the tunnel bottleneck. The tunnel bottleneck means the narrowest part of the tunnel. Then we have a maximum bottleneck radius, average length of the tunnel, and average curvature of the tunnel, or accompanied by corresponding standard deviations. In analysis folder, which is one of these 3 folders you get in the outputs, there are several files. The first one describes the tunnel characteristics in individual structures, and so that you can, for example, plot the bottleneck radius, so the narrowest part of the tunnel, across all the structures. In here we call it snapshot from MD, and you see there is quite a lot of variance in the bottlenecks, coming from about 1 angstrom to nearly 2.5. Then there is data to analyze overall tunnel geometry, so-called profiles. It is it's basically the tunnel radius plotted along the tunnel length, and here we can see the profile of the same tunnel in 2 different structures. The blue one just being short and quite wide, and the red one is obviously longer, more structured, and in most cases it's narrower, or in all cases it's narrower. Further, if you use in your configuration file the parameters in italics, in this presentation I will use italics to indicate that it's a parameter of the configuration file. So if you use compute tunnel residues, yes, the analysis folder will also contain the 2 text files, which lists the atom and residues lining the tunnel. And similarly, if you specified compute bottleneck residues, yes, you will get a list of bottleneck residues. These files look something like that, so you get the information about chain, or protein chain, the residues, then how many times they contributed to the tunnel or the bottleneck, how many times it involves actually the side chain of the residue, not the backbone, and which atom and how many times were involved from this residue. Another parameter you may use is generate bottleneck heat map, yes, which generates the content of a bottleneck heat map folder. And this allows you to study the evolution of bottlenecks in individual structures simultaneously for all detected tunnel clusters. So in the heat map, you then can see 5 tunnel clusters and their respective bottleneck radius, which are actually color-coded, meaning the narrowest bottlenecks are red, about 1 angstrom, and green are the widest bottlenecks. So we can observe the fluctuations of the tunnel bottlenecks. For example, for the first cluster, you see it in these first few structures it's very close, then it opens, close, opens, and so on, so on. In some cases, you see that the other 4 tunnels are really identified only rarely, and they are very narrow. And maybe important to say the gray means that the tunnels was not found in a given structure at all. And similarly, you can use generate profile heat map, yes, a parameter which allows you to obtain the time evolution of a whole tunnel profile. So it means that you have a geometry of single tunnel cluster evolving or changing through the individual structures, the snapshots, And you can see from the beginning of the tunnel to the end on the vertical axis how the radius of the tunnels changes. So you can see that the narrowest part, the bottlenecks, are located in sometimes in quite a different location at the end, in the middle of the tunnels, and you can also notice the differences in overall width of some tunnels and also their length. Sometimes the tunnels are very short, sometimes very long, and it's all considering just a single representation of a single tunnel. Now coming to the second folder you will have as a result from Caver, this is PyMol. Running the generated script, you will find there, will provide you with the visualization of the tunnels. So using the view timeless PI, you get all tunnels across all analyzed structures mapped on a single structure, which is very useful for just the overview where the tunnels are in the different structures all together and compare them. It's also good for the clustering setup, but we will get to that later. Then there is view Py script, which requires safe dynamic visualization to be set to yes. And this shows you the dynamics of the tunnel, but only on the single static protein structures. And if you use both, say dynamic visualization yes and generate trajectory, you can use the last script, which is view trajectory PI, and that actually shows you the tunnels in the content of their respective structure. So you have dynamics tunnel in protein dynamics. And finally, related to the PyMol, if you set compute tunnel residues yes, you will get atom PIs, and this allows you to visualize the tunnel lining atoms of respective tunnels. And in case you will have a lot of structures, a large ensembles, you can use VMD instead of PyMol, which can handle a larger number of structures. And all you have to do is specify a path to your VMD executable, and then you work similarly to PyMol, either with all tunnels in one structure or analyzing the dynamics of tunnels and proteins in once. So now we are getting to the last part, maybe the second half of the webinar, which will deal with 4 critical settings and some common pitfalls in using Caver. And basically it's all about where to start searching for the tunnels, how narrow the tunnels can be, what is the same tunnel and what is already different tunnel, and finally where are the ends of the tunnels. So to define a starting point for Caver calculation, you can use one of the following parameters with ID of atoms you want to use, or residues, or directly supplement the coordinates of your starting point, which is probably the hardest. You should put an effort to position your starting point as close to the center of the cavity as possible. And to help you with this task, Caver performs some optimization of the starting point, which is governed by 2 parameters, where max distance simply means how far the optimized starting point can be from your user-defined starting point, and the desired radius basically defines or represents the minimal distance from the optimized starting point to the surface of any protein atom. So we are saying, okay, I want to put the optimized starting point into some large cavity or in the center of the cavity, something like that. And in most cases, the default setting, as it is, should suffice to obtain reasonable tunnels, but sometimes there is a common pitfall in an improper positioning of starting point. And here on this slide you can see a protein in gray. There is actually a cavity from where you would like to find a tunnel. In red you have residues selected to define starting point, and the user defined starting point is in green. And since the starting point is located further from the cavity, further from the cavity, the optimization algorithm cannot translate the starting point into the cavity, and therefore the initial portion of the tunnel in blue in here is very thin. And if you have the minimal tunnel radius larger than this very narrow part, you will not even get any tunnels. The solution is either to really use the residues symmetrically surrounding the cavity, which is this situation, and therefore you will get your user's defined starting point close by the center of the cavity, and optimization just assures that you have a really excellent position and you have a very nice tunnel. Or the second solution would be to increase the maximal distance for the optimization. In this case, it would be from 3 to 6 angstroms, and that would allow the optimization to actually find very similar starting points to this proper manual settings. However, this second approach, increasing this max distance, could be a source of other possible errors. And it's especially with the maximal distance, so we will have the same situation. If the maximal distance is too large for our system, the starting point can be even displaced out of the relevant cavity. In this case, it would go basically outside on the surface of the protein, and you will have only a single sphere before you actually reach bulk solvent. This is easy to detect, so there's not such a problem, but sometimes you have more cavities in the protein, and it can just jump to some other cavity which is not relevant. There is not binding, anything happening, so you will get tunnels leading from very different positions. So beware of that. The second critical consideration is about minimal radius of a tunnel that caver can search for or should search for, and this can be defined by the probe radius. It's in angstrom, and it's important to note that so far what we've seen that the radii of calculated tunnels are generally smaller than would be expected from the size of transported ligands. And here on the graph, there is actually the fluctuation of the narrowest portion of the one tunnel through very short MD simulation, 10 nanoseconds. And you also can see radii of the same tunnel in this dotted lines in the same tunnel obtained from 2 crystal structures, and you see that the differences can be quite significant. So we advise that you should search for narrower tunnels before you conclude that there is no tunnel in this region of your interest, or that after some change the tunnel is missing in some homologs or in mutant proteins or something like that. Next, now we are getting to the clustering of the tunnels, which is used by caver to distinguish which tunnels are actually just less important alternatives of the same tunnel, and which are relevant branches or even different tunnels and should be retained in the analysis. The level of the details provided on the tunnel branching is governed by a clustering threshold parameter, and the second parameter, one tunnel in snapshot, defines if we should analyze alternative tunnels or only the most relevant tunnels for each structure. So if we set no, it allows us to see all tunnels, which is very important for the setting of this clustering threshold. But on contrary, the option cheapest should be used for any other analysis, not to noise your data and visualization with this alternative tunnels, which mostly are not relevant. They are narrower than the cheapest tunnel, and everything important will happen through this cheapest tunnel. Again, mostly the basic settings should give you at least good starting point for your analysis, if not already perfect solution, but you can sometimes come over some suboptimal clustering threshold for your system, and this can unfortunately hide the presence of some relevant tunnels from you, or in a better case just result in a really high number of irrelevant branches of the tunnels. On this slide, you can see a cluster which would obviously represent a single tunnel, maybe with some infrequent branches. And okay, yeah, branching. And if you would use this one tunnel in snapshot no and visualize all tunnels, then you will see that in some structures there were actually tunnels heading even in the opposite direction than the original tunnels, and that would indicate that we used a really too high threshold for the clustering, which actually clustered all the tunnels in the structure into the one. Fortunately, this is very easy to correct. You just lower the clustering threshold, and you will see that if you go to 4.5, you will have 3 nicely defined tunnel clusters. And possibly, if we want to study some tunnels even in more details, their branches, so we can decrease the threshold further to separate the branches into individual clusters. So you may see like 2 tunnels going from same position to basically same position at the surface. There you have one large tunnel and some small branch nearby the surface. You can study that if you want to focus on some particular region. Additional decrease would lead to something like this messy picture, which is capturing just some subtle differences, and should be rather considered as a noise. So if you see something like that, really overlapping clusters, not well defined, you should, on the other hand, increase the clustering threshold. And the last consideration which can help you with ideal settings of your calculation is related to where should the tunnel end. So, of course, the answer is simple. It should be on the protein surface. In Caver, the surface is defined by 2 parameters, shell radius and shell depth, both in angstroms. And the Caver uses a rolling probe algorithm to define so-called outer surface of the protein, which separates a surface region from a bulk solvent. However, such surface would be very crude. To capture more details like surface grooves, funnel-like tunnel mouths, and so on, Caver employs this shell depth parameter to define so-called inner surface in a distance from the surface of the shell probes. So once you have these 2 surfaces, inner surface, outer surface, you can, or Caver actually search for the tunnels starting from the starting point, and the tunnels are computed to all points on the inner surface. So there you can get many tunnels ending at the inner surface. And then only the single widest tunnel is prolonged from the inner surface to outer surface. This means that the tunnel branching is disallowed in the surface region. And if we consider the default setting of these 2 parameters, they should again be sufficient for most of the system. And there are also some additional phase-safes implemented to handle some small deviation in these parameters from the optimal parameters to your system. But still, sometimes you may have to modify these parameters, and in such cases, you can encounter some problems. When you have relatively wide tunnels, it may happen that the shell radius is too small, and thus you will obtain very short tunnels, as your surface can extend even into the protein cavity. So if you see extremely short tunnels ending somewhere inside the protein, probably you have too small shell radius. You should increase that, and you will see. In other case, you may be missing some relevant branches closer to the surface of the protein, and in that case, most probably your shell depth is too large, and no branching, as in this case, is allowed from the inner surface. So actually, you find only the cheapest, the widest pathway, but you miss the second one. So in such a case, just try to decrease the depth somewhere in here, and you will see that the branches will start to appear. And the last case, you will probably see even with the optimal solution is that you could have some non-optimal ratio between these two parameters, meaning that you have two large shell radius and two small shell depth at once. It can be just a tiny fraction difference that you would need. And in this case, you can recognize that you have to slightly increase the shell depth by occurrence of many tunnel branches, which will be like in this green tunnel, will be branching very close to the surface, and they may even appear as if they are slithering on the protein surface. Mostly they are irrelevant, and you just need to increase the details by the shell depth, so these really small grooves won't be used by the tunnels to explore some new ways on the surface. And this brings us to the end of our webinar, which I would like to finish with a really short advertisement. We are constantly developing also some other software tools which could be useful to members of SBGrid. These are Caver PyMol plugin, which allows you to set up and run basic calculation directly from PyMol. Then there is a Caver Analyst, which is advanced graphical user interface, allowing interactive calculation of tunnels and also their interactive analysis. The Hotspot Wizard web server, which is suitable for automatic analysis of protein structure, including cavity, tunnel detection, functional and evolutionary analysis, and last being the Predict SNP, which is web server suitable for evaluation of disease-related mutation. So that would be all from my side now.
Great. Thank you very much, Jan. If anyone has questions, feel free to send them to me in the chat window, and I will pass them on. I had a quick question about the PyMol plugin. I think the PyMol plugin is probably the easiest way for people to get started with Caver. It's very easy to use, and it's included in the SBGrid PyMol install.
The Caver plugin is probably the first thing you should use anyway, because it's really suitable to set up the starting position. You just make selection and then say, okay, use this selection, this residues to define the starting point. You will immediately see where the starting point is, whether you should change your selection. And then you have basically only these 4 parameters I discussed in this critical setting there, which you have to take care of. So you are not overwhelmed by these many parameters, which can help you slightly improve, but generally you don't need them, especially if you are just starting with your analysis. So definitely Caver plugin is the way to go at this moment.
Yeah, and this plugin does not have the advanced data output. Is that correct? Like the tunnel radius and those sorts of parameters.
Okay, so basically when you run the Caver calculation by a Caver plugin, it's actually calling the Caver free standalone. And in an output directory which you define there are output directories, the numbered output directories created, which contains all this information. Also, you can actually force the plugin to use full config and change other parameters as well. So you can sort of use it as some interface to Caver free. You cannot probably well handle numerous structures from that, but for a single static structure, that's optimal choice for everything. Unless you would like to wait for the Caver Analyst, which should allow you to get all the output data directly into one program, like these heat maps, ready, graphs, plots, or everything there. But that will be soon.
Yeah, and you said that's this summer.
Yeah. We are just finishing the version, so it will be probably very soon released for some beta testing to Caver users.
Right. Great. Well, with that, and if there's no questions, I don't have. Wait, let me see. I do have one. Thank you very much for this webinar on software. Very informative for me. So, and I'd like to thank you too, Jan. Thank you very much for the webinar, and I urge everyone to fire up PyMol and load up the PyMol plugin and grab their favorite protein and do some caving. So thank you very much.
Welcome.